is super important. Transcription is the first step in DNA, or in protein synthesis, rather. So if I want to make a protein, I need to have information on how to make that protein, right? And then I need to physically make the protein. Okay. So the first thing I need to do is get the information as to how I can make the protein. I need to know what the sequence of amino acids needs to be, and that's what transcription is going to get me. Okay. So DNA is just a whole bunch of instructions on how to make proteins. Okay, so the long strand of DNA has some non-coding regions, and then it's got a lot of coding regions that say this is how you make a protein. Okay, we call those regions genes. Okay, now DNA is really long, it's really big, and it's really important, and so it's not allowed to leave the nucleus ever. So if you want to make a protein and you need the information from the DNA, you need to make a copy of it, right? So I would make the jump here, and this is probably going to be weird for you guys because you don't really use encyclopedias because you have the internet, and the internet is much more awesome. But in the olden days, if you wanted to research something, you would start with an encyclopedia because it would give you a pretty concise piece of information, and you could go from there. Okay, but if you go to the library to use the encyclopedias, you can't check out an encyclopedia. You can't say oh, I'm going to take this Encyclopedia Britannica and I'm going to take the letter V home, right? You can't do that because it's got all the information in it and people need to use them, and so you aren't allowed to do that. So if you wanted to use the encyclopedia, you would just pull it off the shelf, and then you'd go over to the copier and you'd make copies of whatever specific pages you needed, right? That makes sense because you only need, let's say I was studying Venezuela, right? I only need the pages on Venezuela. I don't need all the pages on volcanoes or, vuvuzelias or whatever else is in the encyclopedia for V. And so then I would put the book back. So DNA works the same way. I don't want to use the entire strand of DNA every time I need to make a protein, so I'm going to make a copy of the little section that I need so that I can take that around and move it into the cytoplasm. Make sense? All right. So, transcription is the process of making a RNA copy of a DNA template. So it's worth noting here that this term transcription means that you don't change the message of something. So if you wanted to transcribe everything that I'm saying, right, if you wanted to transcribe this class period, what you would do is you would write down every single word that I said. So you'd write down all the ums and the ands and everything like that, right? So anything that I say, you'd actually write down. You'd transcribe it word for word. You just change forms, right? You change from a spoken form into a written form, but you don't change the message, okay? That's the key to this. You don't change the message that's on the DNA. You keep the exact same sequence of nucleotides. You just transfer its form. It used to be in DNA form, and now it's in RNA form. That's what they call it, transcription, okay? The way that it works is, let's say we've got a strand of DNA. Here's my strand of DNA. And on this strand of DNA, I've got lots of nucleotides. I'm not going to draw a lot of nucleotides. I'm just going to draw a few here. But let's imagine that for every one line I'm drawing, there's actually 50 nucleotides because genes are pretty long. Okay. So the first thing that's important is that there is going to be a region on this DNA that's going to be, decently long, maybe like this long. Okay, and that region is going to be known as the promoter. Okay, and the promoter's job is to signal this enzyme called RNA polymerase that is the place where it should attach. Okay. The next part of this DNA, say, is this whole region here. And this part is called the gene. And that gene is going to contain the instructions for how to make a protein. Okay? So, don't draw this right now, but I'm just going to draw this down here real quick. RNA polymerase. So this RNA polymerase, again, don't draw it yet, okay, is going to be floating around inside the nucleus, right? And it's going to be bouncing off the DNA, and it's like, nope, don't want to attach there, don't want to attach there, don't want to attach there. And then it goes ding, 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 here's a promoter, I'm going to attach to the promoter, and then I'm going to move along this DNA, and I'm going to copy this section called the gene, okay? It's important that it attaches here at the promoter, because if it didn't attach at the promoter, let's say it just randomly attached right here, okay, it would transcribe this, but then it would only get half the gene because it attached halfway through the gene. Right? And that would be bad because then it wouldn't make a functional protein, and so that would be not worthwhile. So it attaches over here at the promoter because that signals it that that is the beginning of the gene, and that's where it should start the process of transcription. Okay? So the promoter is a sequence of DNA that signals RNA polymerase to attach. Okay? Make sense so far? All right. So then the gene that's the sequence of DNA that has instructions on how to make the protein. Right. And then the last part is this thing called RNA polymerase. And RNA polymerase sounds similar to something we've already learned about, which is DNA polymerase. DNA polymerase's job was to attach the DNA nucleotides to the growing strand, and RNA polymerase's job is to attach RNA nucleotides to the growing strand. So after this thing has done its job here, it's complementary base paired all these RNAs onto here, and you're going to end up with something that's called the primary mRNA transcript. Okay. The primary mRNA transcript is an exact copy of that DNA. So an exact RNA copy of the DNA template. Okay? In prokaryotes, some prokaryotes, specifically bacteria, the RNA transcript is good to go. It can then be made into a protein. So in bacteria, only minimal modification is required to make a protein. but not the case in eukaryotes. In eukaryotes, that primary mRNA transcript is pretty much worthless. If that thing got transcribed, or translated rather, it would make some nonsensical protein that didn't do anything, okay? And it probably wouldn't get translated anyways. So in eukaryotes, the transcript must be modified. So it's going to get 3 different modifications. The first one is the addition of what's called the guanosine cap, or the G cap. A G cap is added to the 5' end of the RNA. And the purpose of the G cap is that it's going to allow it to leave through the nuclear pores. It's like its ticket to go outside the nucleus. And the second reason for the G cap is it is what signals the ribosome to attach to that RNA. So it allows RNA to leave the nucleus. And it allows ribosomes to attach to RNA. Okay, so 2 pretty important functions there for the GCAP. The other thing that has to get added is called the poly-A tail. So a poly-A tail is added. To 3' end. Okay, now poly-A, that poly means many, A means adenine, so it's a whole bunch of adenine nucleotides that get added onto the end. Okay, why is that important? Well, this part is one of the really important differences between just like knowing what transcription and translation are and understanding how transcription and translation work. Okay, so at any given point in your body, okay, you've got a whole bunch of these promoters, and most of them are blocked. Most of them are not accessible. Okay, and then if there's an environmental condition that changes, your body releases these things called transcription factors, and transcription factors will open up the promoter and they'll make RNA polymerase more likely to attach to them. Okay, that's going to make the body more likely to make this gene. Okay, so you're making primary mRNA transcripts and you're making mRNA to make specific proteins in order to react to specific situations. Let's say, for instance, that I drink a whole gallon of milk, right? My body needs to start producing lactase really, really fast, right? Because maybe normally I don't drink a whole lot of milk, and so my body just produces small amounts of lactase, but I drank a whole gallon of milk, and so I got all this lactose sugar that needs to be broken down, and so my body needs to make a bunch of lactase, and so it's going to start producing that protein, okay? How does it produce the protein? It produces the protein by making mRNA, okay? So that mRNA gets made, it goes out in the cytoplasm. How long do you think I need to make lactase for? Should I make it for the next hour? Should I make it for the next 5 days? It's like for a very short period of time, right? I just need to digest the milk that I drank, and then I probably am not going to have a lot of milk for you know, a fair amount of time after that. So I just want to produce it for a very short time. So when RNA goes into the nucleus, it's going to start being broken down almost immediately. Okay, so mRNA goes into the nucleus, and right away enzymes attach to it, and they start breaking it down. What that does is it makes the lifespan of RNA in the cytoplasm relatively short. Okay, and the cell can control how long RNA exists in the nucleotides by adding this thing called the poly-A tail. So poly-A tail, whole bunch of adenines on the 3' end, and these enzymes, they work by attaching to the 3' end, and you can imagine them like little Pac-Men, right? And they're just chomping away at the 3' end, and they're eating away at that RNA until they get to the gene. okay, until they get to the actual part that's coding. Okay, and once they get to the part that's coding, that RNA doesn't get made anymore because it would make a nonsensical protein, and it doesn't get made. Okay. What this does is it means the longer the poly A tail is, the longer it's going to take for that enzyme to actually eat away at the whole tail and start destroying the gene. So if you have a really long poly A tail, that mRNA might exist in the cytoplasm for a week, right? But if you have a really short mRNA tail, that mRNA might only exist in the cytoplasm for like 5 minutes before it gets broken down by an enzyme. So proteins that are going to be reactionary, proteins that are going to only be required for a very short period of time, are going to need very short poly A tails, and proteins that are going to be used all the time are going to need really long poly A tails, right? It sort of works like a fuse on a bomb, right? So if you were going to, set off a smoke bomb, it's got a little fuse on it because what damage is a smoke bomb going to do? You're just going to like light it and put it down. But if you're going to set off a bunch of dynamite, you don't want to have a little fuse on there because you're going to light it and you're never going to be able to get far enough away from it, right? So you want a much longer fuse so that it lasts a longer time before it actually blows up, right? So we can say for this that the length of the poly A tail determines how long the mRNA exists in the cytoplasm. Okay, so the last thing that needs to happen is it needs to get spliced. So the mRNA must be spliced. And when we say that it needs to be spliced, what we mean is the introns must be removed. Those are non-coding regions. Must be removed. And the exons, the coding regions, must be joined together. I remember the difference between introns and exons because of their prefixes, right? This prefix IN I associate with the word inert, which means that it doesn't do anything. Does not do anything. Okay, and then I associate this EX here with the word expressed. Okay, and when we say something is expressed, that means that it's visible, that means that it's doing something, right? And so it creates a protein. If you have a different way of remembering it, that's fine, but that's the way that I remember it. Okay? So, let's look at how this works. This thing here is going to be my primary mRNA transcript. Okay, and on this side, let's say I've got my guanine, my G cap, and I've got my poly A tail on that end. And in this drawing, my yellow parts are going to represent introns, and my pink part is going to represent exons. Okay, so I have 1, 2, 3, 4 coding regions in this DNA, and I have 5 non-coding regions. Okay, so in order to get this into a working copy of mRNA, I'm going to have to remove this, I'm going to have to remove this, and I'm going to have to remove this. Okay, we'll talk more about these ends here, why they don't need to be removed, but just know now we call them UTRs, which stands for untranslated regions, and it's not important that they get removed. Okay, so this thing is going to get spliced together. Okay, and when it gets spliced together, it's going to be all bonded together like this. And we're still going to have our UTRs on the end. And then we're going to have our poly A tail over here and our G cap over here. And we've got what, 1, 2, 3, 4 exons there. Okay. And so now this thing is ready to be made into a protein because we've taken all the exons, all the coding regions, and we've put them together, and now this thing contains only the coding parts of the DNA, and therefore, or coding parts of the RNA rather, and so therefore it's ready to be made into a protein, okay, because it's not going to have any sort of nonsensical parts. Okay, makes sense? You just take out the parts that don't do anything. All right, this is how we've known it works for a long time. We've figured out relatively recently that there's something else that goes on though, and that something else that goes on is called alternate splicing. Okay, so we used to have this hypothesis that was called the one gene, one polypeptide hypothesis, which means that every gene on the DNA created one polypeptide. Okay, and we know now that that's not correct. That one gene can create a whole bunch of different polypeptides, and the way that that works is instead of making this sequence here, it could also make a sequence where you had a UTR and then a UTR, and then you had maybe only 3 of those regions. All right, you had region 1, region 3, and region 4, or something like that. You get your poly A tail, and your G cap, UTR, and a UTR. Okay? And so because this one has only coding sections 1, 3, and 4, for whatever protein this is, section 2 was considered an intron, and so it got removed. Okay? This is going to make a totally different protein from this. But they came from the exact same region of DNA. They came from the exact same primary mRNA transcript. They just got spliced together in different ways. So this is called alternate mRNA splicing. And that's when we join together the exons in a different order. So exons can be joined together. In different orders to create different proteins. And the important part about being able to create different proteins here is they still come from the exact same DNA sequence. So create different proteins from the same DNA sequence. Anyone have questions on transcription or RNA modification? All right, that's it.